Here is the scenario that happens more often than you think. A pharma company invests 2 years developing a generic peptide. A bioequivalence data looks clean. The formulation team is confident. They file and then rejection. Not because the molecule didn't work, but because they couldn't prove that it was the same molecule. That's exactly the gap that peptide sameness studies are designed to close. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories Canada. Resolve Mass Laboratories stands at the intersection of analytical excellence and complex drug development, helping pharmaceutical teams become critical challenges in peptides, impurities, and regulatory pathways. So what exactly is the peptide sameness study and why does it matter so much? When you are developing a small molecule generic bioequivalence is usually not enough. Prove the drug reaches the bloodstream at the same rate and concentration, you are done. But peptides are different. Structurally, they are far more complex. And here is the thing that catches a lot of teams off guard. Even a single amino acid change in peptide sequence can shift receptor binding, alter pharmacokinetics, and perhaps most critically increase immunogenicity risk. We are talking about patient safety implications from what might look like a minor synthetic variation on paper. That's why both the FDA and Health Canada have put a clear stake in the ground. Before any bioequivalence data is even considered, you must first demonstrate structural identity through a rigorous analytical comparability package. This is the peptide sameness study. Think of sameness study as resting on two core analytical pillars. The first is sequence confirmation. This means 100% amino acid sequence match, no substitution, no deletion, full coverage of both N and N terminus and C terminus. And if the molecule carries any post-translational modifications, those needs to be identified and characterized too. The workhorse technique here is LC-HRMS, liquid chromatography coupled with high resolution mass spectrometry, often paired with MS-MS sequencing after enzymatic digestion. These aren't just confirmatory tools. Done properly, they tell you exactly what you have residue by residue. The second pillar is impurity profiling and is where things can get genuinely challenging in practice. Synthetic peptides carry a fingerprint of process-related impurities, truncated sequences, oxidation artifacts, residual reagents from synthesis. Regulators don't just want to know your impurity levels. They want to see that your pattern of impurities is comparable to the reference product. These require stability indicating HPLC methods, forced gradation studies, and often LC-MS-MS for structural characterization of individual degradants. Getting this right takes experience because what looks like a clean chromatogram can hide sequence variants and only emerge under stress conditions. Now, a question we hear constantly from clients navigating both markets like Health Canada and FDA: how different are FDA and Health Canada requirements? Honestly, closer than most people expect. Both agencies require same analytical foundation, sequence confirmation, impurity equivalence, stability data, and bioequivalence support where applicable. The strategic advantage here is significant. A well-designed analytical package built with both regulatory frameworks in mind from the start can anchor both your ANDA and your ANDA submission simultaneously. We have seen companies cut months off their timelines simply by planning for dual submission upfront rather than retrofitting that data package after the fact. One practical point worth noting here is Health Canada's ANDS requirements explicitly call out validated stability indicating methods. So this needs to be part of your analytical strategy from day one, not the afterthought. Here is the misconception that derails most of the programs than any technical failure. Assuming that bioequivalence data will carry the submission, it won't. Bioequivalence tells regulators how drug performs in a patient, but it says nothing about what the molecule is. Structural identity has to be established analytically. Orthogonal techniques, head-to-head comparisons against the reference drugs, full degradant characterization. Only after that package is in place will bioequivalence data will be meaningful. This isn't a bureaucratic friction, it reflects a genuinely rigorous scientific standard, and rightly so given the complexity of these molecules. So what separates submissions that sail through from the ones that stall? A few things consistently make the difference. Start your analytical planning at project inception, not after synthesis is locked. Apply orthogonal methods throughout: LC HRMS, peptide mapping, HPLC UV, ion chromatography. Each technique sees a different dimension of the molecule. Fully characterize your impurities and degradants, not just identify them, but understand their origin, potential clinical relevance, and align with ICH guidelines from the start. Q2 for analytical validation, Q6B for specification, Q1A for stability. Regulators notice when a submission is built on a solid ICH foundation. At Resolve Mass Laboratory, this kind of end-to-end analytical strategy is what we do for every peptide program, from early characterization through final submission support. The complexity of these molecules demands this kind of systematic experience approach. So here is the bottom line. Peptide sameness isn't a checkbox. It's the scientific foundation on which your entire generic approval rests. Get the analytical strategy right: sequence confirmation, impurity profiling, stability data, orthogonal methods, and both FDA and Health Canada have a clear path to say yes. If you are navigating a peptide generic program and want to stress test your analytical strategy, I'd encourage you to reach out. These are exactly the kinds of complex program worth solving early before they become late stage surprises. Thanks for watching, and if your program demands precision, expertise, and regulatory ready data, contact Resolve Mass Laboratories to support your complex drug development journey. Thank you so much for watching this session, and see you in the next one. Goodbye for now.